Hello everyone, I'm Fan from Gregorian Lab at Dartmouth College, and today I will talk about our recent study, which is to describe the relationships between sequence structure and stability in proteins based on the analysis of tertiary structural motif. So what does that mean? The protein data bank has been the key to understand the protein sequence structural relationships. For example, the statistical distribution of the sequence in the secondary structure has been very informative in either predicting the structure from sequences or design amino acid sequence for targeted structures. However, the secondary structure cannot fully explain the observed sequence structural relationship because you can imagine for any residues in a structure, the secondary structure only capture a small part of the environment for that residue. So we hope to, oh, sorry. So to address this problem, we want to analyze the sequence structure relationship in a more complicated unit, and we want this element be more complicated than secondary structure, but still more simple than the protein domains to focus on the local physical chemical effects. So so therefore, we introduce the tertiary motifs, or terms for short. Terms are usually multiple segments of secondary structures that are associated by multiple segments that are not necessarily close in linear sequence, but associated by the inter-residue contacts. And using a software, a structure search software previously developed in our lab called Master, we can search this geometry in the PDB for all instances with the similar geometries and generate a sequence pattern for this term. So now the question is that is this information meaningful or just noise? So to answer this question, we have some proof of principle analysis. First, we show the application in structural prediction by using term statistics to assess the quality of models in structural prediction. In this framework, a structure was broken into its constituting terms, and each terms were searched against the PDB, and we developed a scoring function to indicate the sequence structure compatibility based on the statistics. And for benchmark, we use the targets in the structural prediction competition CASP. And here, we found the score of models correlate quite well with the similarity between the model to the actual structure. And also on the per residue score, also distinguish the poorly predicted region. As you can see here, if we divided the residues in the dataset into 3 groups based on their score, and here the red-blue colors indicate the accuracy of these residues, and you can see the scores largely recapitulate the accuracy of the residues. So recently, we further showed that the statistics of terms can be used to predict how amino acid mutation affect the stability of proteins. The strategy is that we create a term that covers the position of interest. So the term is a description of the local structural element for this position. And the key is to, and this this fragment will be searched in a PDB, and the key is to compare the relative frequency of the 2 amino acids. And we have promising performance on the public dataset of experimental measures thermodynamic parameters. So in a large dataset, we have a good correlation, and in a smaller dataset, which enables direct comparison to many other predictors, our term method actually outperforms many of other predictors out there. So as a conclusion, we show that enlarged PDB database make it possible to ask statistics questions about more complex structural units, and we define tertiary motifs and search and show that the statistics are physically meaningful, linking the sequence structure and stability of proteins. And we believe our method will facilitating the progress of structure prediction and protein design. And of course, I omit a lot of details here. So if you are interested, please come to my poster L28 in the Monday session. Thank you for your attention.